yesterday. So there's a different type of transformation. So you can have the natural competence, right? That means some bacteria cell, they have the ability to uptake the DNA from the environment. That's natural competence. But sometimes you can also induce the competence that we will discuss later. And then you can have the protoplast transformation. So basically you remove the cell wall and then you mix with the DNA, and then the cell can uptake the DNA, right? So that's protoplast. And then electroporation. So basically you mix the DNA with the cell, and then you apply the high voltage electricity and to shock the cell, and then the cell will uptake the DNA. That's electroporation. Okay, and this is the protoplast fusion, right? The protoplast, you remove the cell wall and mix together, then they can fuse together, and then so they can recombine the DNA. And induce competence. So basically, in the laboratory, okay, the bacteria cell, they don't have the ability to uptake the DNA by themselves. Then we have to induce the competence. To induce the competence, then we have to apply the specific chemical. We use the calcium chloride, which in the next two slides we will discuss the calcium chloride. Then we can change the cell wall property so that the bacteria cell will be able to uptake the DNA efficiently. Okay, and then the microinjection. So this is the gene gun. That basically in the gene gun you have the DNA you put into the probe and then shot into the cell so that the DNA they carry the DNA and then enter into the cell. And this is the microinjection, right? So here's the probe, right? And then you can put the DNA in the probe and then it can enter into the cell directly. That's called a microinjection. And then the induced competence. So basically you soak the DNA in the ice cold calcium chloride concentration, calcium chloride solution, all right, 50 millimolar calcium chloride, and then that can that will change the cell wall property so that that will make the DNA better precipitate outside the cell wall, and then you can apply the heat shock so in this process when you apply the calcium chloride, you can induce the competence so that when you apply the heat shock, the DNA will be able to enter the cell. So this process is induced competence and then transformation. All right, and then of course when we normally perform this process in the laboratory, all right, we have to also do the recovery process after the transformation, after the heat shock. Okay, and so the transformation is the recovery process that we discussed yesterday. That's the reason why we need to have a selectable marker in the vector, because selectable marker can help us to identify which cell uptake the DNA. And during this recovery process, basically those plasmid DNA will produce the antibiotic resistant gene, will produce the antibiotic resistant protein. Okay, and therefore after the recovery process, when we put this recombinant cell into the selective media, then we'll be able to see those cells that carry the recombinant plasmid will be able to survive in the presence of the antibiotic. All right, so that's why the recovery process is important during the transformation procedure. And then later on, you'll be able to distinguish the recombinant cell from non-recombinant cell. Okay, any question? Very good, Neil. Nancy, where are you? Rodana, Sarah. Alexandra, Myra. Good. Okay. So that's the process of a transformation. That first of all, you induce the competence by using the 50 millimolar calcium chloride to change the cell wall property. Yes, I call you Moham. And then after you induce the competence, you mix the cell with the DNA, and then you apply the heat shock so that the cell will uptake the DNA. After the heat shock, the most important step is the recovery process. It is extremely important. This is step that you can distinguish the cell uptake the DNA or not. And later on, you place those cell in the selected media, and then you will be able to identify the recombinant cells. Okay, so that's the recovery process. And then, as I mentioned yesterday, that we will discuss further about the insertional inactivation. Okay, so on the left, this is normal vector molecule. On the right, that's the recombinant vector molecule. So the recombinant vector molecule, basically, you introduce the target gene in a piece of a DNA of interest into the vector, into this target gene, that because the target gene has cloning site. Once you introduce a piece of the DNA into here, it disrupt the whole the target gene, all right? And this phenomenon, this situation, we call it insertional inactivation, because you inactivate this gene product. Okay, and the example of is the first example here is the PBR322. That's the insertional inactivation of antibiotic gene, right? And antibiotic gene. So PBR322, you have both tetracycline and the ampicillin resistant gene, all right? And then in tetracycline resistant gene, you have the cloning site, BANH1, right? If you introduce a foreign DNA sequence here, so you disrupt the tetracycline, all right? Therefore, this plasmid will only provide ampicillin resistant, not a tetracycline. But the original vector has both ampicillin resistant and the tetracycline resistant property. Therefore, introduce a piece of DNA into this BANH1 site, you inactivate the tetracycline resistance. Okay, so this is insertional inactivation of an antibiotic gene. And then to screen, all right, to screen the PBR322 recombinant by the insertional activation of the tetracycline resistant gene, all right? So basically, after you perform the transformation, all right, and then you put those cells in the ampicillin media, and then, so basically after the transformation, so let's go from the beginning. So you have the mixture of the vector, and then you cut the vector with the BNH1. After you cut this vector with BNH1, then you mix with the insert DNA. So some vector will ligate this insert DNA at this position. Some vector will perform the self-ligation, so that you will have two different type of vector in the mixture. All right, one is the original vector, the other one with the insert after the ligation process. All right, and then you have the mixture of these two different type of vector. Then you perform the transformation. After the transformation, now the first step is to screen which cell carry the plasmid. All right, so that you put the cell in the amphicoelin media. All right, and then all the colony that you see on the amphicoelin media that represent every single cell carry either this vector or this vector. All right. And then the next step is to identify which one of these would be this recombinant molecule, not this original molecule. In order to do that, though, then you have to do the replicate plating. All right, replicate plating, transfer those cells from the amphicoelin media to the tetracycline media. All right, transfer to the tetracycline media. Then once you transfer to the tetracycline media, all right, so the cell that carry this original vector will survive, and the cell that carry this recombinant molecule will not survive, so that you will see the colony here. And then these colony are tetracycline resistant colony, which means these colony are these colony carry this vector. All right, and then for those colony disappear in the tetracycline, that would be the colony, the bacteria cell carry this vector. Any question? Nancy. Clarice. Nancy. What is the question? The purpose of a recombinant colony. What do you mean? Nancy, what do you mean the purpose of the recombinant colonies? The recombinant cell, that means they carry those recombinant DNA. So they are the recombinant cell, the recombinant colony. Where's the Clarice? Good. Very good. So this process is very important, okay? Will be on the test. You have to understand this, all right? So this is the insertional inactivation of antibacterial and how you clone the gene and how you identify the recombinant cell. Okay, and then the next example would be the insertional inactivation of the non-antibiotic gene. All right, in this case, would be the LEGZ gene. All right, so the LEGZ gene here we have PAC8 vector. All right, the PAC8 vector that has the ampicillin resistant gene and also have LEGZ gene. In this LEGZ gene, we have the BANH1 cloning site. All right, for the recombinant molecule, you clone a piece of DNA into this BANH1 site. All right, BANH1 site, so that you will inactivate the LEGZ gene expression. But the original vector can express the LEGZ gene. So you produce the beta-galactosidase, all right? And this one will not produce a beta-galactosidase, okay? And so the difference would be the LEGZ gene, when they express, all right, in the presence of S-scale, all right? In the presence of S-scale, that gives you the blue color, all right? And then if it's a recombinant plasmid here, so the LEGZ gene will not be able to produce the beta-galactosidase, and then they will give you the white color. All right, that give you the white color. So for the transformation of the so for this procedure in the beginning, all right, in the beginning, you mix the PAC8, the vector, with the gene of interest, the DNA of interest, after the ligation. So you will have a two group of the vector plasmid. One, the PAC8, they self-ligate, all right, self-ligation, so they don't have the insert. And second group, that with the insert. So you have two population of the DNA. One is original vector. Two, that's recombinant molecule. And then you perform the transformation. So when you perform the transformation, only the part of the cell will update the DNA, all right? And those DNA can be original vector DNA or the recombinant DNA, all right? So now you have three different populations of a cell. Number one would be the cell does not have the cell, they don't have the any DNA molecule. Number two would be the cell that uptake the original vector DNA. Number three would be the cell uptake the recombinant DNA. All right. In order to distinguish these three different populations, all right. So then you can after the transformation, you can put those mixture into a plate, a medium that contain the ampicillin and also S-scale. All right, the ampicillin plus S-scale. Therefore, the colony you see that will indicate those are the cell carry DNA. Doesn't matter the original DNA or recombinant DNA, because either this original DNA or recombinant DNA, they all carry the ampicillin resistant gene. So that's why they can survive and produce the colony. And then you check the color. If it's a white color, that means that will be this recombinant DNA molecule, because they disrupt the expression of beta-galactosidase, and therefore they cannot produce the beta-galactosidase, so that they cannot cleave the S-scale, and therefore their colony be white color. And then if it's a blue color, that means the cell carry this original vector DNA, because they produce the beta-galactosidase, and this beta-galactosidase will be able to cleave the S-scale and give you the blue color. All right, so that's how you screen the recombinant DNA molecule, all right, to distinguish the difference between these two.